Good morning, everyone. Today I'll be talking about my work on development of computational models for the notch signaling network. The work was actually done as a part of my PhD thesis when I was at Professor David Schaefer's lab, and it was published last year in computational biology. Professor Schaefer's lab is interested in various different signal transduction pathways in the context of cells, and understand how they regulate cell proliferation and difference in order to ultimately harvest them for therapy for neurodegenerative disease. Various complex cell environment where the very different other cell types, extracellular soluble factors, and all of the inputs the cell, which are received through the receptors on the surface of the cell and are transduced within the cell through a cascade of signal transduction pathways. These signal transduction pathways can be very complex and can involve feedback and feedforward loops and crosstalk between the pathways. And these ultimately result in regulation of gene transcription within the nucleus of the cell that culminate into an output of as a cell behavior of the cell. So in order to harvest cells for therapy, it is important to understand how the input signals relate to the output cell behavior. And one such signaling pathway that we were interested in is the notch signaling pathway. Notch is an evolutionarily conserved signaling pathway which is involved in various cell fate decisions during the development of an organism and also in the adult organism. Notch is a 300 kilodalton transmembrane protein which undergoes contact dependent upon binding of ligands delta and jagged. And this binding initiates a transduction cascade where the intracellular domain of notch is cleaved and translated to the nucleus to initiate transcription of downstream target genes. And one of the very known target genes for the notch signaling network is this Hess family of genes. And interestingly, this signal has been known to function in a variety of ways during the development of an organism. And the notch pathway is known through lateral inhibition specified nerve cells of epithelial cells, while on the other hand, it works as an oscillator to additionally, during the specify one type at the expense of another. Sort of linear pathway work in these and have these various different roles. That's the question that we were interested in, and in order to answer that, we decided to model the signaling network. So, looking more closely at the regulation of the Hess genes through the notch delta signaling, we'd see that the Hess promoter here, I'm showing that in green are the transcription factor binding sites for the Hess protein itself, and they're called N boxes. And the red are transcription factor binding sites for a transcription factor called RBPJ kappa. And this is generally a transcriptional repressor. So this is the surface of the cell where the notch protein is expressed. And upon contact with the neighboring cell where the delta or jagged protein is expressed, as I said before, it undergoes cleavage at the cell surface, and the intracellular domain, or NICD as it is called, can translocate inside the nucleus, bind to RBPJ kappa, and turn it from a transcriptional repressor to a transcriptional activator, and turn on expression of the Hess, which is then transcribed and translated into protein. And this protein can then go back into the nucleus and bind its transcription factor binding sites, the N boxes, and repress transcription. So this whole network formed back loop right here. There have been previous attempts to model this system and various behavior aspects of notch signaling. And we'll look at the notch delta contact dependent signaling to explain explain, and there's another set of models that explain this next gene transcriptional circuitry, and they explain the work. Reveals something interesting. This work is composed of primarily 3 different genes: Notch, HES, PJ kappa. And if we look at the transcription, the promoters of the Notch, RBPJ kappa, we see that these also have HES and RBPJ kappa. So in essence, are very tightly bound in a regulatory network where each of them are the other two. So we in its entirety, and as of these arrows, the schematic diagram is a different. In the interest of time, I'm just going to briefly differential equation system for HES part of the model. For HES mRNA, the HES protein in the cytoplasm and the HES protein in the nucleus. We have a localized model. The nucleus and the cytoplasm are, of course, different. And we've incorporated transcriptional and translational delay times in the model. Processes are inherently, they take a long time. All the mRNA and protein decay kinetics are assumed to be first order. Just very quickly, I like some equations. Here we have the Notch mRNA, the full-length Notch protein, and also the ISCB, the nucleus, and the cytoplasm. But importantly, this parameter here, the K del P, defines the signaling strength of delta binding to Notch. So this term here describes formation of the NICD upon binding of Notch to delta. And we'll be later on in some of the results. And one more thing that I'd like to explain in the model is the transcriptional term. So if we go back here, this rate of formation term is defined as the transcriptional term for the mRNA. And the transcription of genes are just defined as a sum of the signals of the basal transcription signal and induced signal. And the induced signal is the sum of the probabilities of the promoter in being in a particular state by the transcriptional rate in that state. For example, if this promoter has 2 transcriptional factor binding sites, one particular state of the promoter could be that there's nothing bound to any of these sites, or there's 1 transcriptional factor bound to site 2. So the induced signal in this case would be the probability of the promoter being in this state multiplied by the rate of transcription when the promoter is in this state, and the sum of all the probabilities of all the possible states. So now that we have the model in place, what we wanted to do is first look at how a cell responds to an input delta signal, a continuous input delta signal. So here I'm showing a cell is at steady state, and at a given time we give it a continuous delta signal, and these 2 lines represent 2 different strengths of the delta signal, represented by this term K del P. This is essentially the K del P term. So you see there's a lower input delta signal, the HES1, and the output of the system is quantified by the expression of HES1. So in the case of a low input delta signal, the system or cell still expresses low levels of HES1, and we call this the off state. However, as we increase the delta signal by about 10-fold, now the concentration of HES1 shows an exponential increase, and it plateaus out at a steady higher expression level of HES1, and we call this the on state. So we see that the system now exhibits bistability, where as we increase the level of the input signal, the cell can now reside in, express a higher steady state level of HES. So what is the switching point of this? In order to decipher that, we did an experiment where we progressively increase, and then we find that at about this level of delta initiation, the cell transitions from an off state to on. Now the interesting thing is, if we go backwards, so if we have a cell which is expressing high levels of Hess, and we progressively decrease the delta strength, it comes back to the off state, but the switching point is now at delta signal strength. So what does this mean biologically? Oops. In an ideal and deterministic world, a switch is just a step function. But we know that biological systems are noisy. So for biological systems, this would be a problem because any stochastic noise within the system around this switching state would just mean that the cell keeps switching back and forth between the on and off state. Whereas if a cell exhibits hysteresis, as I just showed you that this notch delta signaling system does, this provides the cell with some noise filtration capabilities, and it enables it to switch its state in an unambiguous fashion where it has this much leeway to, in the input signal to switch its state. So far, the results I've shown you have all been for a continuous signal of the delta. However, during development, there are various instances where the delta signal is only transient. So how does the system respond to a transient delta signal? And here we've done the analysis in two different ways. First, we looked at the effect of the duration of the signal. So this plot here shows the effect of the Hess expression as a function of time when the input signals are varied from 10 minutes to 3,000 minutes. And we see that the expression of S1 increases progressively to about 800 minutes when the input signal is about 800 minutes. And after that, giving the signal for a longer duration of time just increases the time that the system is in the on state, but it does not further increase the expression level of Hess1 within the cell. Similarly, an analysis on the signal strength of the delta shows us that as we increase the signal strength of the delta, the Hess1 expression levels increase up to the final on state, but at that, it only increases the duration until which the system is on. So in short, we see that for transient delta activation, either give a moderate signal for a long amount of time or a very high and short pulse signal. So these signals are provided for only about 100 minutes. So a high intensity short term signal to be able to turn the state on and keep it on for an amount to actually differentiate into its desired state. So this is all very interesting. We have shown how a cell can switch its state upon notch delta signaling, but we would also like to know how the model is, how sensitive the model is to the various parameters in the model. So first parameter that we looked at was obvious, the half-life of NICD. The half-life of NICD is generally pretty long. It's in the order of hours. And this is the reason why we see the prolonged expression of Hess1 within the cells, even in a transient delta signaling state. But as we decrease the half-life of NICD in the model, we progressively decrease the Hess1 expression levels in the cells. However, if we go down below a threshold level, now the cell does not even turn on. It cannot express higher levels of Hess1 within the cell. So this means that this shows the level of delta activation required in order for the cell to transition from the off state to the on state as a function of the degradation constant of NICD, or inversely the half of NICD. So the higher the degradation constant, the lower the NICD. So these transitions are highly dependent on the half-life. So what does this mean biologically? In a normal cell which has prolonged half-life of NICD, we have high levels of expression. Turn on, we have high levels of Hess1 expression. And Hess1 is a transcriptional repressor which has been known to repress various oncogenes. So we have cell in a normal state. However, if we have a shortened half-life of in a cell due to some reason, its in turn not turn the switch on and we would have expression levels, which may in turn turn some of the oncogenes into a tumor cell. And in fact, we have precedence. We know that integrin-linked kinase reduces NIC stability, and this would in turn decrease its half-life. NIC levels have in fact been observed in various cancer patients or tissue types. So we further looked at some other parameters in the model, such as the equilibrium binding constant of an ICD to RBP, transcriptional rate of others, which I would refer you to the paper if you're interested. But the majority of these parameters, we saw that the off, even though they're dependent, even though they're dependent on the parameter values, the qualitative nature is the same, and it only shifts the bistable and off to on transition. However, we found a very interesting parameter in our model, and this is the repressive constant of HES1. So far, in the model, we've used a repressive constant of 0.3, which is based on some previous studies. But as we decrease this repressive constant here, we see that the maximal HES1 expression states also progressively decrease. But this only happens in the range from 0.3 to about 0.1. What happens if we go further down? So if we go down about 10, have a repressive constant of about 0.03, so a repressive promoter, we see that the system is no longer a switch. Now it's transitioned into being an oscillator. And this kind of starts to elucidate how this notch system can function as a switch in one context and an oscillator in another context. And I had previously shown you that some studies modeled the HES1 network to elucidate the oscillation part of this signaling network. And all of those studies actually used an RN box value of 0. So they had used a totally repressive model for this system. So this is very consistent with their models as well. So we did a very comprehensive study of the whole parameter range, and we found that our system, which initially was in this range and was acting as a switch, can transition from being a bistable switch to a very brief period of monostability and then down to an oscillator, depending on this one single parameter value, which is the repressive constant of HES1. So I think this kind of begins to elucidate how this single signaling system can work by tuning its parameter values as in context as a switch as well as an oscillator. So in summary, we've modeled the notch signaling network encompassing the 3 key genes and looked at the activation of the HES1 gene. The model indicates that the network exhibits bistability and can function as a switch in certain ranges of the parameter. And the transient activation of the network is sometimes also sufficient in the biological context to act as a cell-fate switch. And then finally, the repressive constant of HES1 is a very key parameter in the model that can be tuned to qualitatively change the behavior of the system from a switch. That we also did some stochastic model the biological network, and there is some stochastic noise. If you're interested, because I didn't have time to go into at that, I'd like to acknowledge Professor Dave Schaefer, who was a great mentor for me during my PhD, and Colin Archer, who helped out with the stochastic model that, again, I didn't have time to show here. And the funding was provided by NIH and the Helen Wells Neuroscience Institute, and I'd also like to thank ISCB, Department of Energy, and NSF for providing partial funding for me to attend this conference and present this work here. With that, I'd also like to thank the committee for giving me the opportunity to present this work here, and thank you all, and take any questions. Oh, sure. So the stochastic model basically showed that if the signaling, it undermines some of the bistability of the network. So if the system undergoes transients signaling for a very short amount of time, the switch properties still hold. But for longer periods of duration, there is some stochastic variability, and some cells can spontaneously switch their state. But by and large, I think what we found was the results of the deterministic model did hold.